Good day, this is Professor Will, CBMD PhD. Today's Tuesday, July 6, 2021. It is 3:38 and 15 seconds, p.m. Eastern Standard Time. Today's research, Epsilon variant. So let's hop to it here. Epsilon variant mutations contribute to COVID immune evasion. Date July 6, 2021. Source, University of Washington School of Medicine, UW Medicine. New studies reveal an unprecedented mechanism behind the loss of antibody neutralization against this pandemic coronavirus variant of concern. 3 mutations in the Epsilon coronavirus spike protein dampen the neutralizing potency of antibodies induced by current vaccines or past COVID infections. The mutations give this coronavirus variant of concern a means to totally evade specific monoclonal antibodies used in clinics and reduce the effectiveness of antibodies from the plasma of vaccinated people. And to better understand the exact immune escape strategies at work here, the scientist visualizes various infection machinery to see what is different from the original configuration of the pandemic coronavirus and what the implications of these changes are. For several years, researchers have been exploring the molecular conformation and infection mechanics of SARS-like coronaviruses. They also examine how antibodies attempt to block infection mechanisms and how variants come up with new dodges. Their latest data shows that the Epsilon variant relies on an indirect and unusual neutralization escape strategy, according to the researchers. A molecular clock analysis timed the emergence of the precursor to the Epsilon variant to May of 2020 in California. By summer of 2020, it had diverged into its B1427/B1429 lineages. COVID cases from the variant increased quickly, and the variant soon became widespread in the United States. It has now been reported in at least 34 other countries. And to learn more about the characteristics of the Epsilon variant, the researchers tested the resilience against the Epsilon variant of plasma from people who were exposed to the virus as well as vaccinated people. The neutralizing potency of the plasma against the Epsilon variant of concern was reduced about 2 to 3 decimal 5-fold. Like the original SARS-CoV-2, the variant infects target cells through its spike glycoprotein, the structure that crowns the surface of the virus. The researchers found that the Epsilon mutations were responsible for rearrangement in critical areas of the spike glycoprotein. Electron microscopy studies showed structural changes in these areas. Visualizing these mutations helps explain why antibodies had difficulty binding to the spike glycoprotein. One of the 3 mutations in the Epsilon variant affected the receptor binding domain on the spike glycoprotein. This mutation reduced the neutralizing activity of 14 out of 34 neutralizing antibodies specific to that domain, including clinical stage antibodies. The other 2 of the 3 mutations in the variant affected the N-terminal domain on the spike glycoprotein. The researchers used mass spectrometry and structural analysis to find that a part of the coronavirus N-terminal domain was remodeled by these mutations. The signal peptide cleavage site was shifted in the NTD antigen supersite, and a new disulfide bond was formed. This resulted in total loss of neutralization by 10 out of 10 antibodies tested specific to the N-terminal domain and the spike glycoprotein. The scientists believe that uncovering mechanisms of immune evasion, such as the newfound mechanism based on signal peptide modification, is as important as variant surveillance through RNA sequencing. And together they know such efforts can help to successfully counter the ongoing pandemic. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.